And that leads me into the last of the three products we're going to talk about today. The Xvie XO concentrate is a multi-signal concentrate for follicle reactivation, and it is an in-clinic product. What's really exciting about this product is that we have found in proteomic analysis, so we've done full mass spec on all of these products. Every single lot to date has had mass spec on it, and which means I know exactly which proteins and exactly how much of each proteins is in all of our products. We find over 700 unique growth factors and peptides that are shown to signal and regulate the entire hair cell growth, supporting extended anagen growth. And we see that not only are these particular signals vital for hair regrowth, but that the source is very important. And we're going to talk a little bit more about sourcing, but the source is the human amniotic fluid. And the reason why we utilize it is that it has been shown in research to contain regenerative rich cell signaling to support the scalp and the follicle microenvironment. This is a product that is minimally manipulated. It is not culture and expanded. We'll talk about that in a minute. It's 100% natural. It does not contain any antibiotics, no cryopreservation chemicals, no fetal bovine serum or culture media. That has no preservatives and no fragrance. This is for in-clinic use, and so we're very excited to roll it out to our providers that we're working with for their in-clinic use. And a quick snapshot of this particular product, you can see, as I mentioned, those 700 unique growth factors and peptides, hundreds of trillions of signaling growth factor molecules, hundreds of billions of exosomes and extracellular vesicles, naturally occurring hyaluronic acid, and in addition to that, peptides, amino acids, phospholipids, electrolytes, all of the amazing things that we know are a part of human amniotic fluid, and why human amniotic fluid has been used as a regenerative medicine product since the very early 90s, 1900s, not early 90s, but was also used then too. And what's really important to note about utilizing human amniotic fluid as our source is that as we look at the competitors that are out on the market, we wanted to understand what is the best possible source for this type of product for hair regrowth specifically. And so there are other sources out on the market that includes human adult adipose, PRP, which most of us are probably very familiar or most familiar with. There's also adult bone marrow products, human umbilical cord lining products, animal umbilical cord lining products, and then plants and fruit products, different botanicals. What I really want you to take away from this is that what we find in the research is that one, those products that are derived from plants or other botanicals, while they may be great, unfortunately, they oftentimes have been proven in the research to not match the biological function of a human. And so what I mean by that is that the plants and fruits and other botanicals, they, as a biologic source, fall short because they cannot signal with cells the way that a human source can. This often is seen as well on the animal side of the house. And what is most important about all of these products is that they are all what we would call culture and expanded products. These culture and expanded products, unfortunately, are essentially a single cell or a single derived source or donor that then gets grown in a petri dish over and over and over again for many, many passages, oftentimes for years, sometimes longer than a decade, I've seen. And unfortunately, in research and as a scientist, it's well known in the scientific community that we do not continue to culture cells over and over and over again into perpetuity because we know that the more times that those cells reproduce themselves, the more likely they are to mutate. So eventually they will mutate to a point where they are no longer kind of the originating cell that we thought they were. And that's well established in how we understand and utilize cell culture in the lab. And so the other piece is that is really important here about utilizing human amniotic fluid is that it is one of the richest natural sources of regenerative molecules in the world. And we go through a processing technique that basically harnesses all of that, gets rid of the cells. It is decellularized. This is not a cellular product whatsoever. I remove all the cells during this processing. It allows for tissue repair. It's androgenetic. Allows for wound healing, which there's a lot of research behind utilizing amniotic fluid as a wound healing product in peer-reviewed literature. It's antioxidant and it can promote immune regulation and homeostasis, in addition to cell proliferation and more. So when we think about human amniotic fluid, then of course the question is going to come up of donor sourcing. And that is something that I am in the thick of every single day. This is a big part of my job is the donor sourcing. And so when it comes to donor safety and ethics, it is incredibly important to me. All of our product is from full term donor, fully consenting C-section births and entirely healthy births. So I like to give the example of my own journey in life. My first kiddo was an emergency C-section and my water broke. That would not be a good source of our product. But my second birth was a planned C-section and my water did not break. And so instead, if I decided that I wanted to consent to giving my amniotic fluid after the birth or at the birth, essentially what they would have done is I would have gone in to do that procedure. They would cut down to the membrane, use a sterile needle, collect the amniotic fluid sterilely because it is a sterile product to begin with, as long as you collect it sterilely, and then they would send it to the lab. And so what's important here to note is that the donors are not compensated per the regulations of the American Association of Tissue Banks, and they are thoroughly screened per the American Association of Tissue Banks. So imagine all of the screening requirements that are necessary for you to donate a kidney. All of those things are required for you to donate human amniotic fluid for our product too. And so we hit all of those standards, not only with the AATP, but also with the FDA. And processing matters. So perfect segue into, so someone has decided to be a consenting donor. They go in, they give birth. Fabulous, wonderful. We're very happy for them. We receive their amniotic fluid the next day in the mail from just a couple of states over in North Carolina. So all processed within the United States. And then we go through a minimal manipulation ultrafiltration processing technique. And what this does is it allows us to start with the initial product, and through centrifugation and ultrafiltration, we remove all of the cells, and then we are just collecting the extracellular vesicles or exosomes, the growth factors, the naturally occurring hyaluronic acid, and these other molecules that we've already talked about. And we've already profiled each lot one by one, which is very important. This results in a 100% natural product. We are doing so very carefully that it literally takes me all day to make one of these batches of products. And it is, once processed, immediately frozen at -80 and kept at -80. It then goes through rigorous standards of quality, which then leads to about a 3-week quarantine timeframe before we actually ever get to a point of releasing this out to the public. This not only is so important because the amniotic fluid contains specific stem cell types, multiple stem cell types, that then we are deriving not only these extracellular vesicles from, but also all these growth factors and cytokines and other peptides that we've now profiled. And what's really important here, and that I want to hit on one more time, is that this product is not, under any circumstances, considered culture and expanded, which the other products that are out on the market in this arena typically are. And so, what a culture and expanded product is, is one only approved by the FDA for research and not for commercialization. And the reason why is because culture and expanded products use an immortalized cell line, which means a cell that never dies. And quite frankly, another category of cells that never die that we utilize in the lab every day are cancer cells, because they are immortal and they grow constantly. And they can be expanded again and again for years or even decades. And what can happen then, unfortunately, as I mentioned, is that those cells, as they're expanding over time, can then mutate, and that's well established. And unfortunately, those mutations can impact then the product itself, right? What kind of extracellular vesicles are being derived from these cells? What kind of growth factors? Are they anti-inflammatory or pro-inflammatory? And what's the impact to the product? And some culture and expanded products not only because of the nature of growing a cell in a petri dish, can they mutate? But because of the way they grow culture expanded cells, that means that it also contains fetal bovine serum because we have to feed the cell when it's living outside of a body. We have to give it food. So typically it contains fetal bovine serum. We have to give it antibiotics like penicillin, which helps to make sure that the media that we're feeding the cells doesn't become contaminated with yeast or bacteria or similar. Oftentimes, then they're also trying to keep those cells alive, so they have to use something like DMSO or other synthetic preservatives for these particular products. And because there's fetal bovine serum, unfortunately, you can imagine just by the name, fetal bovine serum has a smell to it, especially over time. And so, oftentimes, they will add a fragrance to this particular product in order to reduce that scent. In addition to that, the secondary part of a lot of these culture and expanded products, not all, but a good portion of them, also lyophilize their product, which means they take their extracellular vesicles or their exosomes and they freeze dry them. They turn them into little raisins. And what's interesting is we're getting more and more third-party research showing that these particular portions of the products, once you freeze dry them, while they may still contain and hold their spherical shape, that they're no longer as bioactive as they were before. And what I mean by that is that on the outside of exosomes or extracellular vesicles, there are specific signaling factors that are all attached to the outside of the exosome. And that is what connects to another cell and tells the cell, hey, I want to tell you a story. I want to give you these growth factors. I want to give you these peptides, for example, and then allows that cell to say, oh, yeah, I recognize you. I see that, and I'm going to accept this, these messages that you're bringing to me, these growth factors, for example. But if the lipid bilayer is in any way, shape, or form destroyed, or those signaling factors have been impacted, which now we have research that shows that they are being impacted by lyophilization, they're not able to signal as well as they should, which is why we do not lyophilize our product. Our product is received in the clinic on dry ice. It is kept at an ultra low freezer temperature for the entirety of its existence until you go to use it in your clinic. For exactly that reason, we know that extracellular vesicles are most bioactive when they have been properly preserved in an ultra low temperature. And unfortunately, because some of these products have then not only culture expanded, but then lyophilized their product, they see themselves as being able to be room temperature or room stable. And unfortunately, what we see is that if you're lyophilized, it's likely that you're not as bioactive anymore, as I mentioned earlier. And if you're not lyophilized, that those exosomes are degrading very rapidly. We have our own shelf-stable data at multiple temperatures, and there's also third-party data behind this to show, yes, exosomes are not shelf-stable, which makes entire sense, right? Like, of course, this bioactive product wouldn't be something that's shelf-stable that you can keep at room temperature. So we're losing stability very, very quick in these products if they're not kept at an ultra low temperature over time. In addition to that, of course, I want to mention that not only one of my favorite things about working for Express A being the fact that I get to let all of the creative juices flowing with these incredible experts, and the product R&D is so rigorous. But more than that, as a scientist who's making products that I want to make sure that I feel so comfortable with them that I would utilize them with my own children, is that every single one of our quality standards goes through a third party. You never have to take my word for it. It is entirely third party tested. We are manufactured in an FDA registered facility, fully AATB and FDA compliant. We do third-party testing screening for all serology. So there's a blood sample that is taken from the donor upon birth. That's sent to Eurofins for full testing and of all relevant diseases per the American Association of Tissue Banks and the FDA. And then in our final product, that also gets sent out to Eurofins for sterility, endotoxin, mycoplasma, and additional tests that we do for every single lot per the FDA as well. As an added layer, which is super fun for someone very nerdy like me, we do one more step, which is that we do every single lot of our product out for protein analysis, so nanoparticle tracking to understand what our extracellular vesicle concentrations are for every product. But we also send this out for third-party proteomic analysis, which means I know and have mass spec on every single one of my products. And It's complex, right? So as I mentioned, if we have over 700 different growth factors and peptides, when we look at these, this is just a small snippet, obviously, of those 700. Not only do we know which growth factors and peptides exist within our product, we know how much, general concentration. And that ties this all together beautifully to talk to you about that up and down regulation. Because XV contains this rich natural source of signaling growth factors in the product itself, we then have tested this alongside these other cell lines to see, okay, what is the impact as it relates to those upregulated growth factors and biomarkers that promote the anagen phase and maintenance and continued hair growth, and what are we downregulating that would reduce the suppression and the miniaturization of the particular follicle. We, of course, did additional testing on this, not only proliferation assays and protein assays, but again, a wound healing assay, and we see complete closure. So even faster wound healing and higher cell proliferation, still within that sweet spot and window to show like, yep, we are increasing proliferation, not to a rate that is concerning, but we get full wound healing in just 36 hours in an in vitro assay with this particular product. And so when we compare this product, what's really fascinating and what's fun is that because I have proteomic analysis and nanoparticle tracking data on every single lot I've ever made of XV, that means that I can go into the literature and pull all of the peer-reviewed information that I have about PRP. And so what we did is we did a comparison of these two, all of the literature that exists for PRP in comparison to all of the data that we've gotten from third-party analysis on our product. And what we found is that about one treatment of XV equals or exceeds about 6 treatments of PRP. And so that's based on not only the nanoparticle count of our particular product, but also the bioactivity of our product. And so it has been shown in peer-reviewed literature, and we're currently in the thick of a histology research study to prove this out and study this even further, is that human amniotic fluid has a longer sustained release of bioactive molecules over about a 10 to 12 week time frame versus PRP, which is about a couple of weeks. And this is well established in the literature. And so as we compare these then, that leads us to these particular treatment indications that we're looking at in the clinic specifically. So as we look at actual case studies then, this is an example of that type of treatment where we are seeing for this particular patient, a 56-year-old female with female pattern hair loss, that one treatment of Xvie scalp microneedled at day 0. You can see here after a month, you can see the first growth that has started. And then at month 2, look at that impact in this person's hair growth. It's really incredible. And as we've looked at all of this data and research, typical recommendation, if you will, typical protocol will be for one treatment of Xvie in clinic microneedled every quarter. So every 3 months or so is a really great time frame for these individuals to allow for the hair growth to occur and then for them to come back and be assessed and determine the next treatment. This is another case study. This is a 49-year-old female. And here I really also want to point you to those temples up here. So you can see the hair growth. And this is just one treatment of Xvie on day 0 that you see there of baseline with microneedling. Another example, again, at just 30 days post microneedling of the scalp with Xvie treatment. One of the things I'm very excited about is some of our founding team actually really hones in on hair loss as it relates to those really, really hard cases, right? So these are some of the hardest cases as it relates to hair loss. For example, this is a 43-year-old African-American female with proven CCCA. And look at this growth over the course of just a 4-month time frame. Really, truly incredible. And these are providers who literally, when we started this, said, nothing treats this. Nothing can help. Nothing gets hair growth for these particular patients. And at first, we were even like, do we even go through treatment and attempt to tackle these cases? And the answer was yes. And we're so excited to see the preliminary results that are coming out that are going to go towards full-blown research study publications. We have several IRB supported studies in Xvie as well. one more case study for you because I just love looking at these pictures. And then we'll shimmy into questions here shortly. This is a 32-year-old African American male, and same type of situation. This is at just 1 month post treatment with Xvie via microneedling.